Phylum Porifera. The invertebrates, or invertebrata, are animals that do not contain bony structures, such as the cranium and vertebrae. The simplest of all the invertebrates are the Parazoans, which include only the phylum Porifera. The sponges. Figure 1. Parazoans, beside animals, do not display tissue-level organization, although they do have specialized cells that perform specific functions. Sponge larvae are able to swim. However, adults are non-modal and spend their life attached to a substratum. Since water is vital to sponges for excretion, feeding, and gas exchange, their body structure facilitates the movement of water through the sponge. Structures such as canals, chambers, and cavities enable water to move through the sponge to nearly all body cells. Morphology of sponges. The morphology of the simplest sponges takes the shape of a cylinder with a large central cavity, the spongocoel, occupying the inside of the cylinder. Water can enter into the spongocoel from numerous pores in the body wall. Water entering the spongocoel is extruded via a large common opening called the osculum. However, sponges exhibit a range of diversity in body forms, including variations in the size of the spongocoel, the number of osculi, and where the cells that filter food from the water are located. While sponges, excluding the hexactinellids, do not exhibit tissue layer organization, they do have different cell types that perform distinct functions. Pinacocytes, which are epithelial-like cells, form the outermost layer of sponges and enclose a jelly-like substance called mesoil. Mesoil is an extracellular matrix consisting of a collagen-like gel with suspended cells that perform various functions. The gel-like consistency of mesoil acts like an endoskeleton and maintains the tubular morphology of sponges. In addition to the osculum, sponges have multiple pores called ostia on their bodies that allow water to enter the sponge. In some sponges, ostia are formed by porocytes, single tube-shaped cells that act as valves to regulate the flow of water into the spongocoel. In other sponges, ostia are formed by folds in the body wall of the sponge. Choanocytes, collar cells, are present at various locations, depending on the type of sponge, but they always line the inner portions of some space through which water flows. The spongocoel in simple sponges, canals within the body wall in more complex sponges, and chambers scattered throughout the body in the most complex sponges, whereas pinacocytes line the outside of the sponge. Choanocytes tend to line certain inner portions of the sponge body that surround the mesoil. The structure of a choanocyte is critical to its function, which is to generate a water current through the sponge and to trap and ingest food particles by phagocytosis. Note the similarity in appearance between the sponge choanocyte and choanoflagellates, Protista. This similarity suggests that sponges and choanoflagellates are closely related and likely share a recent common ancestry. The cell body is embedded in mesoil and contains all organelles required for normal cell function. But protruding into the open space inside of the sponge is a mesh-like collar composed of microvilli with a single flagellum in the center of the column. The cumulative effect of the flagella from all choanocytes aids the movement of water through the sponge, drawing water into the sponge through the numerous ostia, into the spaces lined by choanocytes, and eventually out through the osculum, or osculi. In the meantime, food particles, including waterborne bacteria and algae, are trapped by the sieve-like collar of the choanocytes, slide down into the body of the cell, are ingested by phagocytosis, and become encased in a food vacuole. Lastly, choanocytes will differentiate into sperm for sexual reproduction, where they will become dislodged from the mesoil and leave the sponge with expelled water through the osculum. The second crucial cells in sponges are called amoebocytes, or archaeocytes, named for the fact that they move throughout the mesoil in an amoeba-like fashion. Amoebocytes have a variety of functions, delivering nutrients from choanocytes to other cells within the sponge, giving rise to eggs for sexual reproduction, which remain in the mesoil, delivering phagocytized sperm from choanocytes to eggs, and differentiating into more specific cell types. Some of these more specific cell types include collencytes and lophocytes, which produce the collagen-like protein to maintain the mesoil, sclerocytes, which produce spicules in some sponges, and spongocytes, which produce the protein spongin in the majority of sponges. These cells produce collagen to maintain the consistency of the mesoil. The different cell types in sponges are shown in Figure 2. In some sponges, sclerocytes secrete small spicules into the mesoil, which are composed of either calcium carbonate or silica, depending on the type of sponge. These spicules serve to provide additional stiffness to the body of the sponge. Additionally, spicules, when present externally, may ward off predators. Another type of protein, spongin, may also be present in the mesoil of some sponges. The presence and composition of spicules, spongin, are the differentiating characteristics of the three classes of sponges. Figure 3. Class Calcarea contains calcium carbonate spicules and no spongin. Class Hexactinellida contains 6-rayed siliceous spicules and no spongin. And class Demaspongia contains spongin and may or may not have spicules. If present, those spicules are siliceous. Spicules are most conspicuously present in class Hexactinellida, the order consisting of glass sponges. Some of the spicules may attain giant proportions, in relation to the typical size range of glass sponges of 3 to 10 millimeters, as seen in Monoriphis chuni, which grows up to 3 meters long. Physiological processes in sponges. Sponges, despite being simple organisms, regulate their different physiological processes through a variety of mechanisms. These processes regulate their metabolism, reproduction, and locomotion. Digestion. Sponges lack complex digestive, respiratory, circulatory, reproductive, and nervous systems. Their food is trapped when water passes through the ostia and out through the osculum. Bacteria smaller than 0.5 microns in size are trapped by choanocytes, which are the principal cells engaged in nutrition, and are ingested by phagocytosis. Particles that are larger than the ostia may be phagocytized by pinacocytes. In some sponges, amoebocytes transport food from cells that have ingested food particles to those that do not. For this type of digestion, in which food particles are digested within individual cells, the sponge draws water through diffusion. The limit of this type of digestion is that food particles must be smaller than individual cells. All other major body functions in the sponge, gas exchange, circulation, excretion, are performed by diffusion between the cells that line the openings within the sponge and the water that is passing through those openings. All cell types within the sponge obtain oxygen from water through diffusion. Likewise, carbon dioxide is released into seawater by diffusion. In addition, nitrogenous waste produced as a byproduct of protein metabolism is excreted via diffusion by individual cells into the water as it passes through the sponge. Reproduction. Sponges reproduce by sexual as well as asexual methods. The typical means of asexual reproduction is either fragmentation, where a piece of the sponge breaks off, settles on a new substrate, and develops into a new individual, or budding. A genetically identical outgrowth grows from the parent and eventually detaches or remains attached to form a colony. An atypical type of asexual reproduction is found only in freshwater sponges and occurs through the formation of gemmules. Gemmules are environmentally resistant structures produced by adult sponges wherein the typical sponge morphology is inverted. In gemmules, an inner layer of amoebocytes is surrounded by a layer of collagen spongin that may be reinforced by spicules. The collagen that is normally found in the mesoil becomes the outer protective layer. In freshwater sponges, gemmules may survive hostile environmental conditions like changes in temperature and serve to recolonize the habitat once environmental conditions stabilize. Gemmules are capable of attaching to a substratum and generating a new sponge. Since gemmules can withstand harsh environments, are resistant to desiccation, and remain dormant for long periods, they are an excellent means of colonization for a sessile organism. Sexual reproduction in sponges occurs when gametes are generated. Sponges are monoecious, hermaphroditic, which means that one individual can produce both gametes, eggs and sperm, simultaneously. In some sponges, production of gametes may occur throughout the year, whereas other sponges may show sexual cycles depending upon water temperature. Sponges may also become sequentially hermaphroditic, producing oocytes first and spermatozoa later. Oocytes arise by the differentiation of amoebocytes and are retained within the spongocoel, whereas spermatozoa result from the differentiation of choanocytes and are ejected via the osculum. Ejection of spermatozoa may be a timed and coordinated event, as seen in certain species. Spermatozoa carried along by water currents can fertilize the oocytes born in the mesoil of other sponges. Early larval development occurs within the sponge, and free-swimming larvae are then released via the osculum. Locomotion. Sponges are generally sessile as adults and spend their lives attached to a fixed substratum. They do not show movement over large distances like other free-swimming marine invertebrates. However, sponge cells are capable of creeping along substrata via organizational plasticity. Under experimental conditions, researchers have shown that sponge cells spread on a physical support demonstrate a leading edge. For directed movement, it has been speculated that this localized creeping movement may help sponges adjust to microenvironments near the point of attachment. It must be noted, however, that this pattern of movement has been documented in laboratories, but it remains to be observed in natural sponge habitats. Section summary. Animals included in Phylum Porifera are Parazoans because they do not show the formation of true tissues. Except in class Hexactinellida, these organisms show very simple organization. With a rudimentary endoskeleton, sponges have multiple cell types that are geared toward executing various metabolic functions. Although these animals are very simple, they perform several complex physiological functions. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.